Welcome to the nervous system. Great job so far. You got through the bones, you got through skeletal, you got through muscle. So start working on muscle and nerve and really pay attention when you're doing muscle to the action potential and the use of neurotransmitter and the excitable membrane, depolarization, repolarization, hyperpolarization. And I might not have mentioned hyperpolarization in that particular topic, but we did it when we talked about the cell, excitable membranes. So the nervous tissue, the nervous system, these cells are called neurons, the specific parenchyma of the nervous system, neurons. And those are the excitable cells because their membranes are very excitable to sodium entering, cause depolarization. So neurons conduct electrical impulses. It happens very fast. So when you hear nervous system, think electrical, right? Think quick, fast acting. Neurotransmitter, depolarization, action potential, repolarization. And the neurons also have supportive cells called glial cells. These are not excitable cells, right? Glial cells, and you'll learn 6 different glial cells, 2 in the PNS, the peripheral nervous system, and 4 in the central nervous system. So these are like kind of like other cells which are non-conductive. They just support, and you'll see what their functions are. We'll go through each one of those functions one at a time for glial cells. Okay, again, understanding neurotransmitters, which we'll talk about these chemical messengers that not hormones, they're not hormones, they're in the synapses of the nervous system. Like you learned one of them in muscle, and that was acetylcholine, right? It was very excitatory to the muscle membrane, and it bound to a receptor. So certain drugs, pharmaceuticals, work either to be agonists for these neurotransmitters, which increase their activity or mimic their activity. Some of them are antagonists, which stop their activity and try to reverse their activity. So that's kind of an important slide. I never thought of it, but talking about that at this point, but that is kind of important. How the nervous system could be excited or inhibited to get some type of specific job done. Okay, so it's again, the brain is part of the central nervous system, is really the master control. So I'm gonna go forward and say brain here. Because outside of the brain and the spinal cord, and even the spinal cord, so I could even say outside of the brain, the nervous system is basically straightforward, nuts and bolts wiring, like an electrical system, but the brain is much more complex. And what we know about the brain at this point is not a lot. So we're going to have electrical signals and chemical signals. The electrical signals are all about the ions, sodium, potassium, calcium, chloride. So they're about ions. The chemicals are the neurotransmitters. Important little facts here. And it's very fast, lightning fast, and very specific and pinpoint in the job it's trying to accomplish. Like something simple as exciting a muscle to contract, right? The contraction of a muscle. And it's usually an immediate response. There is a slight delay, but I'm talking tenths of a millisecond of delay, depending on the neurons involved, the targets, and where the signal is coming from. So this is really important now. You're going to have sensory input, right? Something that's coming into the nervous system. So input has a term called afferent. Let's get that out of the way now. Afferent means coming in. Like in this case, it's coming into the central nervous system. And the central nervous system, we're going to go through this again. The central nervous system is easy to remember. It's only 2 parts. The brain and the spinal cord is central nervous system. Everything outside of the central nervous system, outside of the brain and spinal cord, is considered peripheral nervous system. So information like input, like whatever, it could be pain, sight, temperature, All these senses, special senses and somatic senses, come into the central nervous system to be processed, and that's called afferent, into the central nervous system. So of course, input has to be sensed, and these are very important. Receptors, like your photoreceptors in your eye for special sense, your pain receptors on your fingertips and other places, thermal receptors for temperature, mechanical receptors for vibration, like in your ear, for hearing and balance, chemical receptors on your tongue for the chemicals that are in certain foods. Gustation is taste. Olfactory receptors, which are chemical receptors for the molecules in the air that you smell. Like olfaction is smell. All right, so these receptors are bringing in information about internal, which could be like blood pressure, how much oxygen is in my blood, how much carbon dioxide is in my blood. And external is everything else. All the external changes, like change in dark to light, or the pain versus fine touch, those kind of examples. The opposite, not the opposite really, but number 3, I want to skip to this one, because the motor is the opposite in direction. This is called efferent. And this is going out of the CNS. Remember, the CNS is the central nervous system. And the central nervous system is the brain and the spinal cord. So the motor output coming from the brain, right? Mostly it starts in the brain, just like the cerebral cortex, where we talked about muscle contraction. So it's output, efferent, and there's a target place, and it's not a receptor because this is motor now. So the target for efferent neurons is going to be an effector organ, right? So this is important for somatic muscle contraction. So you have to know what kind of muscles we're talking about, effectors, because you could have skeletal muscle, which you know a lot about. And then you have smooth muscle. You have cardiac muscle, right? Those are the 3 muscle types. And then you have the glands. So these are the effectors for the motor part of the nervous system. So this one here, skeletal muscle, is voluntary. Therefore, the somatic nervous system will affect muscle contraction of skeletal muscle. So somatic involves voluntary, like volition. And also that you could have sensory, which would be cognitive. So I would put that up here, like somatic sensory is what you cognate, which you realize what you're smelling or seeing or feeling. So somatic is cognitive. But in motor, somatic motor, it's voluntary. So that's the somatic portion of the motor system. And then we have what's called autonomic. Nervous division, or nervous system division. And that affects these 3. So smooth muscle could be your blood vessels, walls of your blood vessels, right? The tunica medias. And then you have your digestive tube walls, which are smooth muscle. You have your reproductive and respiratory and other parts that are lined with smooth muscle. So smooth muscle is affected autonomically, involuntary, of course. Autonomics, like automatic, involuntary. Cardiac muscle is strictly the heart, right? You know the heart. And glands, like the adrenal gland, sweat glands, and other glands are affected more by the autonomic nervous system, involuntary, automatic. So many times, like especially somatic, and remember efferent, if you want to remember motor output, remember it's going to an effector organ, and that's a target organ too. So in between, and these are usually in the CNS only, you have these, what's called integration. So you could have sensory neurons, motor neurons, and you have integrative neurons, or sometimes they're called association neurons or interneurons, which you'll see. So the interneurons are basically between sensory and motor, kind of integrating. And sometimes they're between sensory pathways too, between two sensory neurons. But for the most part, they're in the in-between sensory and motor. Like a reflex is an excellent example of how an interneuron, integrative neuron works. When you have sensory input, if I hit you in the knee with a hammer and the patellar ligament, right, patellar ligament, and you extend your leg, the integration of the output is done in the spinal cord, CNS, through interneurons, and the output is the extension of your knee. It's the motor output. So the middleman is the interneuron or integrative neuron. So these are really the functions, right? And we'll go through it. This will be described many times. It's very repetitive, right? So this is looking at sensory versus motor output. So the blue is your sensory neuron, and that's afferent. So in this case, this is a special sense. You're looking at the glass of water, or Tito's, whatever it is. If you're Jeremiah, it's Tito's probably. So you're looking at the glass of water, and that's the input through light, through your eye, through your pupil, to your retina, which is the receptors. And the retina receptors, the rods and cones, are basically a direct pathway to the sensory neuron or sensory input. Now here is where the integration happens in the CNS. The CNS is the brain and spinal cord. So the green neuron is an interneuron, which associates what you're seeing. And then, of course, you think about, once you see the glass of water, you think about picking it up. So that, in that case, you need a motor neuron. So basically out here is PNS. This is outside the brain and spinal cord, right? These neurons. So the motor output is efferent out of the CNS. In this case, it's muscle contraction. It's somatic muscle contraction, which flexes the elbow via the biceps brachii muscle to lift up the glass off the table, possibly bring it to your mouth. Okay, so sensory, integration, and motor. And it gets more complicated, of course, but to know those three functions right off the bat. So to break it down, the most basic way to break this down, and learn this first, the CNS, the central nervous system, is the brain and spinal cord. Remember all the bones you studied, and they're all in the exam, like the parietal bone, the bones, and the occipital bone, and frontal bone, temporal bone. They all protect the cerebrum lobes, and they have the same names, like frontal lobe and temporal lobes, parietal lobes, and then the occipital lobes, because the brain has two hemispheres, the cerebrum does anyway. So the brain and spinal cord is protected by your vertebral column. Remember the vertebral canal, right? The vertebral arch, vertebral foramen, right? That goes down the middle of the spinal column, housing the spinal cord. All right, so the central nervous system, both the brain and spinal cord, are integration centers, control centers. Remember when we did feedback mechanisms? The control center, most of the time, not all the time, but most of the time is the brain, especially a place called the hypothalamus, which we'll learn when we do the CNS specifically. That's in the brain. Right, so it could interpret sensory input, no matter what it is. It could be somatic, touch and temperature and all that, or it could be a special sense, right? And it dictates through integration the motor output, right? But this is all being controlled in the brain or the central nervous system. The PNS is everything else, the peripheral nervous system. It's everything outside. So you'll see it in a picture, and we'll do this in lab in a few weeks. You'll see the spinal nerves exit between each one of the vertebrae. There's like 31 pairs from C1, C2, all the way down to the coccyx, basically. And you'll see the spinal nerves are on both sides, and they basically sending information and bringing in information from those areas. So I'm saying nerve here. Now a nerve is a bundle of neurons. So a nerve contains both sensory and motor, and some autonomic too, which you'll learn. So that's the difference between a neuron and a nerve. A neuron is just one fiber within the nerve. A nerve is a bundle of many neurons. And again, that'll be explained again. Right, so the spinal cord is in the CNS, but the spinal nerves, all 31 pairs from C1 all the way down to the coccyx nerve, are peripheral. They're outside the spinal cord, the nerves, the actual nerves. Cranial nerves, there's 12 of them, 12 pairs, like there's 31 pairs of spinal nerves. And then there's 12 pairs of cranial nerves. And again, you're going to have to learn these cranial nerves. There's 12 of them, and they're bilateral. And some are motor, some are sensory, special sense. Some are both motor and sensory. Some are somatic, and 4 of them are autonomic. So when we get there, we'll get there. But know this, the nerves themselves, cranial nerves, are peripheral nervous system, PNS. All right? So that's the general breakdown of central versus peripheral nervous system. So make it easy on yourself. Learn the central nervous system. It's only 2 things. The brain, which has many areas that we need to talk about, but the largest part of the brain is the cerebrum. And that's all those lobes that match up with the cranial bones. The cerebrum, which has 2 hemispheres, by the way. Then the spinal cord is just that middle part that's encased in the vertebral column that is in the vertebral foramen. Cranial nerves are off of the brain. They come off the brain, which you'll see, but the nerves themselves are peripheral, and there's 12 of those, 12 pairs. And there's spinal nerves, 31 pairs that kind of branch to each side. Like you see all this big network of spinal nerves called the brachial plexus right here, because that goes into the arms. Like again, moving your arms is very intricate all the way down to your fingers. So you're going to need a big plexus here. So you have the cervical plexus, cervical plexus is up here, and then brachial plexus. Then there's no plexus for thoracic nerves. And then you have a lumbar and sacral plexus, because now we're dealing with the legs and all the abdominal contents. Thoracics are pretty straightforward because you basically just have the intercostal muscles, which you'll learn next 2 weeks in lab, what the names are and all that. I'm not going to talk much about ganglia right now because it has to do with autonomic. And we have to learn the neuron first. So ganglia, I'm going to hold on for later. Okay, because it kind of confuse you now. Just stick to the basics. So far, all you have to know is the central nervous system versus the peripheral nervous system, and what you see on this page besides ganglia. Okay, the PNS, peripheral nervous system, has 2 divisions, and it's broken down even further. So now we're in the peripheral nervous system, and these are the sensory, which is afferent, because it's bringing the information into the CNS, right? From where? From receptors in the skin, right? Receptors in the muscles and joints, right? So skin receptors, you might remember a bit of that from chapter 5 in the integument, right? Your Pacinian corpuscles, your free nerve endings that are basically are receptors. Don't forget the receptors have to belong to these sensory fibers. Now fibers, you see the word fibers that your book likes to use, Pearson, here. Fibers really just mean neuron, and a part of a neuron called axon, which we kind of talked about when we did muscle, right? The axon is that long fiber. Skeletal muscles have more Proprioreceptors. Now, proprioreceptors are mechanical receptors for certain things like stretch. Right? They're mechanical receptors. And kinesthetic means movement. And that's what's in the joints, the kinesthetic receptors for movement, especially in those synovial joints that you learned, right, that you're learning now. And those questions are going to be really straightforward on the exam 2B, which will open up Friday morning on joints, but mostly synovial joints. But you need to know the classifications. I'm not sure if we talked about receptors back then, like the Golgi tendon organ or the muscle spindle joint kinesthetic receptors. I'm not sure if we really talked about that, so we're going to leave that for when we talk about reflexes. So somatic sensory is all like the skin and muscles and all that part that we cognate and we have voluntary control over. Now, visceral is going to be more autonomic sensory fibers. So from the viscera means like organs. You know, you get some sensation from your stomach when you're full, or from your bladder, urinary bladder, when it's full, right? Or somebody has, extreme example, somebody has ischemia in their heart or heart attack, you know, myocardial infarction, they get pain from the viscera. So viscera is not somatic, viscera is organ, right? And organs, lungs, kidneys, all those areas have sensory fibers. We don't talk too much about the sensory part because there's so much motor to talk about. So motor, now here we go. Motor is coming from the CNS, and that's efferent. So get those terms down, right? And they're going to an effector. And the effectors are the three muscle types and glands. And it depends, and somatic nervous system, right? So you have two divisions of the motor part. You have the somatic nervous system, and this is really important right here. The effector of the somatic nervous system is only one place. The motor, it's skeletal muscle. You have to get these terms down. Somatic nervous system, motor, right? Motor has an effector, and that's the target. That's the area that's going to do the work once the nervous system sends that efferent signal from the CNS out to the PNS. So somatic is motor is skeletal muscle, and we learned all about that. You got to put all that together because you're going to have a departmental final, and you got to make sense of this. So we have to review this. Okay, the other division is the autonomic nervous system. And get this now, I'll tell you now, the effectors for the autonomic nervous system, effectors, are smooth muscle, like I told you before, and where smooth muscle is your blood vessels and tubes. Cardiac muscle is your heart. And glands. Remember this, really important, because it gets very difficult when we talk about the autonomic nervous system if you don't capture this now. So the autonomic nervous system has two divisions. One is sympathetic, which is fight or flight, increase heart rate, increase blood pressure. Speeds everything up pretty much, physiologically. And then the other division of the autonomic motor, this is all motor still, is parasympathetic. And this is rest and digest. Slows everything down, slows heart rate down, decreases blood pressure, decreases respiratory rate. So you're digesting when you're in parasympathetic stimulation, and you're not digesting when you're in sympathetic. You're more worried about getting blood to your brain quickly and getting blood diverted to your muscles, skeletal muscles, to run away, right? So fight or flight. So if the bear is attacking me, eating my Chick-fil-A out on Route 10, then I'm going to go into sympathetic stimulation. So I have to decide if I want to fight the bear or if I'm going to run away. Of course, the answer is clear. So the blood has to be diverted to my skeletal muscles in case I want to fight, but also to my brain in case I want to run away, right? So or means you got to make that decision, and your brain helps you do that. So before I saw the bear at Chick-fil-A, I'm in parasympathetic stimulation. I'm rested. My digestive tubes are working fine because I'm in parasympathetic stimulation. But once I see the bear, come across Route 10 looking for my spicy chicken and my waffle fries, then I'm going to go into sympathetic stimulation. Okay, back to the somatic nervous system. Now we're talking about skeletal muscle, right? Efferent. And this is motor. Somatic nerve fibers. Let's call it what it is. It's your somatic motor neuron and the axon going towards the muscle. Remember the axon terminals that contain the neurotransmitters. So this is motor and it's efferent. And it's somatic because the target effector is skeletal muscle. And it's voluntary, volition, conscious thought to control the contraction of skeletal muscles. I told you, I don't know how this happens. What happens in my motor cortex of my cerebrum to start an action potential in a motor neuron, upper motor neuron, that ultimately makes my muscles contract. But you know how it all happens. We went through the muscle contraction in great detail. So again, that's what you have to worry about for the next exam. Muscle contraction is very complicated, and the steps of muscle contraction. Autonomic now, more about viscera, right? Viscera, your organs, the walls of your blood vessels. So the effectors are here again. Smooth muscle, which is not just in your blood vessels, it's in the other tubes like digestive, reproductive, respiratory, right? Cardiac muscle is your heart, right? Remember the heart. Like that, Natalia? And then glands, multiple glands, right? Especially the adrenal gland. When we talk about sympathetic, we'll talk about that only to more autonomic specifically. So, of course, this is involuntary. You're not using your cerebral cortex here, right? And this is a function. It's not just a reflex. I mean, there are autonomic reflexes and there are somatic reflexes, like the reflex I mentioned about hitting somebody on the patellar ligament with a hammer to get knee extension is a stretch reflex, but it's a somatic reflex. An autonomic reflex is when your blood pressure drops and your receptors for pressure in your blood vessels send a signal or a sympathetic signal to increase your blood pressure. So it's more about your viscera, right? So the word viscera means like your organs, not your skeletal muscle. So it's involuntary. So two divisions of your autonomic motor, and this is motor, is sympathetic, fight or flight, and parasympathetic, rest or digest. And they're kind of like evil cousins, like a Shakespearean tragedy. We have the agonist and antagonist working against each other. So just to give you a little heads up, you know the effectors now. You know sympathetic and parasympathetic have effectors of smooth muscle, cardiac muscle, and glands. So the neurotransmitter that's released to the effector for each of these. Now, sympathetic releases what's called norepinephrine as a neurotransmitter. Right, that's what's in your EpiPen, right? Epinephrine, to increase your blood pressure in case your blood pressure drops during anaphylactic shock. So norepinephrine, and this is to the effector. You really have to remember this. This is going to the effector, which is cardiac muscle, smooth muscle, or glands, 99% of the time. So that's the neurotransmitter, norepinephrine. Norepinephrine. For parasympathetic, it's going to be acetylcholine to the effector. to the effector, right? There is pathways in between, because acetylcholine is used a lot in the autonomic nervous system, and not just for parasympathetic. It's used for sympathetic as well, but I'm talking about the neurotransmitter that's released to the membrane receptors of the effector. And the effector, again, could be your glands, your heart, or your walls of your vessels and tubes. It's worth repeating over and over, believe me. So they really don't work together very often. Maybe they get a little friendly during their reproductive process. But other than that, they're pretty much opposite. Nice little flow chart here. So take a look at this, starting with the CNS, right? The CNS and the peripheral nervous system, which is the PNS, right? And what they do. So blue lines, that blue arrow is afferent coming into the CNS from the PNS. The red is the efferent going out of the CNS to the CNS. I'm sorry, from out of the CNS to the PNS, and then out to an effector, right? To an effector. So the peripheral nervous system has two divisions right off the bat. Again, don't get confused, go slow, right? The sensory and motor, right? Integration mostly takes place in the CNS. That's why I told you what sensory and motor was first, before I went to integration. So PNS has two parts: the motor going out of the, out towards the effector. Remember, efferent, effector. Sensory coming in, right? And somatic and visceral sensory, and it comes from receptors, which bring information into the CNS ultimately through here, afferent. So the motor has more branches, as you can see. So the two parts of motor efferent division is somatic versus autonomic. Somatic's all about skeletal muscle, right? Skeletal muscle as an effector. Autonomic, cardiac muscle, smooth muscle, and glands. So somatic versus visceral sometimes is a good way to think about it too, because visceral means organs and involuntary. Somatic is more about skeletal muscle, which is voluntary. Makes sense, right? Because organs, remember the hierarchy? We started with subatomic particles, went to atoms, to molecules, to organelles, to cells, to tissue. And we put all those tissues together and got organs. So the organs have muscle tissue. And that's what motor is using as an effector. So now we break down the autonomic nervous system into two branches: the sympathetic division, fight or flight, and the parasympathetic, slowing things down, rest and digest. Now, if you exercise, right, when you exercise, you immediately go into sympathetic stimulation, sympathetic motor. You want to increase your heart rate, increase your blood pressure. It's completely appropriate. So really, I don't need a bear chasing me on Route 10. It'll bother me because I won't get to eat my Chick-fil-A. But in this case, when you exercise, you employ the sympathetic division of the autonomic nervous system. Remember, autonomic, ANS. So ANS. Right? This is somatic nervous system. I don't like to put SNS, but when you get to know it better, you can do that. And here's your CNS, which controls everything. And then your PNS is all the below. All this yellow is PNS. How much is going on out here? Right? So ANS is part of the peripheral nervous system. But everything ties into the CNS, which you're going to learn when you do the next chapter, or the chapter on the CNS and the autonomic nervous system. You'll see. So this kind of memorization, nuts and bolts, like is always part of any anatomy and physiology. You have to get all those buckets of information down before you can move on to the more complicated concepts. It's true. Okay. So I mentioned before, the neuron is what's called the parenchyma, the functional cell of the nervous system, the neurons. But then we have these supportive cells called glial cells, or neuroglia. Neuroglia is just another coverall term for glial cells. Now, these are smaller cells, but I'll tell you right now, there's more neuroglial cells than there are neurons in the central nervous system because they need a lot of support. And we'll go through each one of those. And we'll talk about the neuron, which we really have to get to sooner or later. So when you look at histology, like in the first chapter, really, in the histology chapter, with the tissues, when you looked at the slides of the neuron, or we looked at on mastering, we showed you on the PowerPoint, you saw what the neuron looked like, and you can see nuclei in between that happen to be nuclei of glial cells. So we'll get a closer look at that. So here's some memorization now. Here we go. So now this is the brain and spinal cord, CNS. And these are the supportive cells, also known as glial cells in the CNS. So this one's kind of cool because it's called astrocyte because it looks like a star. So this is one of the glial cells of the brain and spinal cord. And this is an intimate relation with blood vessels. It has to do with blood. So when you hear astrocyte, which is not a blood cell, but it has a relationship with the capillaries. So it helps form, get this, helps form the blood-brain barrier. So I'll tell you this, barrier. The brain is very selective on what it allows into its tissue. So the astrocyte is kind of a go-between between the blood and the brain. It's very selective, and we'll find out about that. Microglial cells are phagocytes, or phagocytic. Phago means to engulf, right? Cells. Again, none of these are neurons, they're all glial cells. So this is kind of like the garbage man of the CNS that kind of engulfs bacteria or virus or cancer or sometimes just faulty proteins that are involved in the brain. Ependymal cells are important for making what's called CSF. And this is cerebrospinal fluid. And this is important because there's kind of like receptacles in the brain. They're called ventricles and subarachnoid spaces that carry this fluid. And most of the reason that we have cerebrospinal fluid in the CNS is to protect the brain, really, to give it a little bit of a cushion. I mean, there are some fluids in there. There's some sodium, there's a slight bit of glucose, some lipid, but still, most of the time, this fluid is to insulate and cushion the nerve tissue, like the neurons of the brain, and the spinal cord as well. Oligodendrocytes, only in the CNS now. These are all CNS, only in the brain and spinal cord. This is the cell that makes what's called myelin. So myelin is a lipoprotein. It's mostly lipid, actually, that wraps around axons of neurons to speed up conduction. And this is what gives the brain tissue and spinal cord tissue what looks like a white matter. Like you hear gray matter and white matter. So white matter is the myelinated axons, for example. So when you hear white matter, it's mostly about the axons that are myelinated with this myelin. And we'll see more of that. So these are the four. You have to memorize them and basically what they do. We'll go through them again. But these are the four glial cells of the CNS, supportive cells, not neurons, not conductive. And by the way, these cells are mitotic. Even in the PNS, more in the PNS actually, which means they go through a lot of cell division. They're all mitotic, all of them. Now a neuron, the nerve cell itself, is amitotic. So when you see an A in front of a word, sometimes like a word mitotic, it means it's not there. It's decreased or it's gone. Like a red blood cell is anuclear. It means it doesn't have a nucleus. So a neuron does not undergo cell division, nuclear cell division. Once it develops, of course, when you're developing as a fetus, it's going to undergo some mitosis and develop into the brain that you're born with. And even that brain that you're born with doesn't go through a lot of mitosis after, say, a year or two. And technically, your brain is not fully developed until you're like 22, 23 years old, especially your cerebrum. Depends on where in the brain, right? But neurons, basically the neurons you're born with are the neurons you're going to keep forever. So you have to take care of those, of course. And you have very many trillion neurons. But it's the pathways that they create is what develops in the brain, right? The connections between other neurons that makes you cognitive things and produces memory. So the neurons themselves do not undergo mitosis. Of course, there's research about some parts of the brain has neurons that may undergo mitosis, so which would really be helpful for people who have horrible brain conditions like Alzheimer's disease or Parkinson's disease or amyotrophic lateral sclerosis, also known as Lou Gehrig's disease, multiple sclerosis. It would really be helpful to be able to use neurons that undergo mitosis to build new pathways that are gone in people with those kind of conditions, especially Alzheimer's. So the ependymal cells, the glial cells really are what support the neurons and give them chances like this. So astrocytes, we'll go through it again. I gave you, just think of blood-brain barrier, right? They're highly branching. These are like foot-like processes, right? They cling between the neuron, the endings, of course. Synapse will tell you what that means, and the capillaries. So this is what's going to form that blood-brain barrier. Astrocytes are very integral in forming that barrier, which is important, but it can be a nuisance when you're trying to treat certain conditions of the brain and you can't get medications in because of this blood-brain barrier. So there is some support, and you see what they look like. You see these legs that brace the neurons. So this is the key that'll play a role in exchange between capillaries and neurons. Remember, capillaries are blood vessels where exchange happens, right? In arteries and veins, you don't get exchange of fluids and nutrients, gases, but in capillaries that happens, right? So again, it's something to do with the development of the brain, of course, right? Control the environment, and that's basically the blood-brain barrier. And respond to impulses through neurotransmitters, and we'll get into that. Of course, they influence neuronal function. They all do. And important, it's about information processing in the brain. So they may have something to do with some memory issues here. But again, a lot of this is still under research. But this is what we know about the connection and the blood-brain barrier. So you hear astrocyte, think blood coming into the brain. So that's a nice picture of an astrocyte. Again, here's your neuron back here. It's usually yellow. Here's a capillary. And you see the astrocyte forms like this Spider-Man web right on there to secure and stabilize the blood vessel to the neuron. And of course, this is a cartoon. And it's a lot tighter of a junction between these two, between the astrocyte and the capillary. And it's the most abundant glial cell in the CNS. Same thing, right? Microglial cells is the garbage man, right? Phagocytized microorganisms like bacteria and a virus and other debris like faulty proteins. And we always have faulty proteins. We're always phagocytizing proteins and DNA and RNA that's within our cells and nuclei. So the byproducts of some of these mutations get cleaned up pretty quickly by microglial cells. And they do their job. They do their job, okay? And look at this. When there's an injury, they're probably going to increase inflammation. So they kind of have a immune response when you think about it. And they'll bring in other cells, immune-like cells. Microglial cells are kind of like branchy, like they call this dendritic arms between the neuron, and in between is the extracellular space, which is some fluid. We're going to have a lot of those faulty proteins running around, and the neuron has cytoplasm, which is the inside of the cell. The fluid inside of the cell is the cytosol. Like this is a nucleus of a neuron, and it's in a part of the neuron called the cell body, which you'll see. So this is the nucleus of a glial cell. In this case, specifically, it's the microglial cell. I don't think we'll ever have pictures of this, but it's good to know what they look like when you're trying to name their function. Again, so the microglial is a immunity or defense cell, phagocytosis. Pendymal cells, first thing you think of is CSF. First of all, pendymal is only in the CNS, right? Brain and spinal cord. Right? And they have different shapes. They're not transitional. They just have different shapes depending on where they are in the CNS, basically. And sometimes they have cilia to move the CSF, right? Remember, cilia is about movement, where the microvilli or villi is about absorption. So cilia is movement of substances along a lumen. In this case, it's the ventricles of the brain which carry the CSF, or the subarachnoid space, which you'll learn later, is the area within the spinal cord and brain where the CSF flows as well. So these line the cavities. So this basically forms a brain CSF barrier. Because the CSF comes from the blood, and the ependymal cells kind of take the blood and they have the machinery that manufactures this cerebrospinal fluid. And then the cerebrospinal fluid will flow through the CNS for a while, like a day, and then it'll be reabsorbed back into the blood. And then we get a fresh input of CSF from the ependymal cells. Here's ependymal cells, tight junctions here. Remember, this is the ventricles, or we call this the ventricle. That's the area in the brain that contains CSF. Or another area is called the subarachnoid space. I'm just throwing this at you just so you know where this is going. All right, and you can see the ependymal cells. Look, and they have nuclei. Here's the nuclei. We're not even looking at neurons in this picture, right? We're just looking at the ependymal cells with their cilia. So the CSF is up in here. So the rest of this is the brain parenchyma of neurons, really. They're not showing it to you, though. So the fluid-filled cavities are called ventricles in the brain and the CNS and subarachnoid space, which you'll see. Specialized structures that are filled with ependymal cells, which you'll see later. Oligodendrocytes, that's a mouthful. And these are the ones that produce and form myelin, which is like a lipoprotein and mostly lipid. So oligodendrocytes have the machinery, DNA and transcription that makes myelin. And the myelin sheath that's going to wrap around the axon, making the conduction, the electrical conduction, faster and more organized. So now this is the CNS, remember. We're still talking about CNS. There's a whole different cell that does this in the PNS. So remember, oligodendrocyte is a CNS. So these are myelin sheaths, so like rings around the axon. Here's your axon fiber, which is called a nerve fiber. Axon is part of the neuron. And there's gaps in between, which I'll show you. These are called nodes of Ranvier. And the PNS, there's different cells that do this, but this is the CNS. And here's the nucleus of the oligodendrocytes. So when a neuron depolarizes, the conduction kind of skips over this lipid, this lipoprotein, and then gets to where it's going a lot faster, like the axon terminal, of course. But this, again, this is in the brain and spinal cord, hence the glial cell is an oligodendrocyte. So that is it for the CNS, naming the CNS neuroglial cells. So now the PNS. And there's two. There's only two now. So the hard part is over. So the two neuroglial cells are satellite cells and Schwann cells, which are also called neurolemmacytes. So again, these are kind of similar to the astrocytes in the CNS, but of course, satellite is only PNS. So they have something to do with maybe like a stem cell for regeneration, as far as healing an injured neuron. They have some combination with the blood, not as extensive as the astrocytes do, though. So we don't really know all that much about satellite cells. Schwann cells are pretty straightforward. Schwann cells, also known as neurolemmacytes, they do the same thing that oligodendrocytes do, except in the PNS. And they look slightly different. So Schwann cells produce and lay down these myelin sheaths. A sheath is like taking a towel, like a really long beach towel, and wrapping it around your arm with many layers. That's basically what the Schwann cell does. It forms this myelin sheath around most of the axons. And it's white. That's why they call white matter white, because it's myelinated axons. So the cell bodies are never myelinated. You're going to see what a cell body is. A dendrite is never myelinated, but the axon is the part of the neuron that's myelinated. And it makes it thicker, of course. And if the nerve fiber is thicker, it's going to conduct even faster. Also has something to do with regeneration of injured nerves. Like if a nerve, I shouldn't say nerve, yeah, I guess I could say nerve, but a nerve is many neurons and axons. But if an axon is injured or kind of torn, the Schwann cells will surround it and try to not regrow, but make the conduction work again. You know, like it's kind of like putting electrical tape around a frayed wire. It's not going to heal the neuron or the axon completely, because it doesn't undergo mitosis. But at least you'll get some help on that from the satellite cells to get the blood in there and help form new cells. As far as Schwann cells, right? Schwann cells are mitotic, right? The neuroglial cells are mitotic. So when a nerve is injured, the Schwann cells could form like a bit of a tube that helps the conduction continue, and it takes time. So here's Schwann cells, and this is a sensory neuron, and I'll show you why I know that later. This is in the PNS. Remember, this is a neuroglial cell. A Schwann cell is in the PNS. So this is a peripheral neuron, of course. And here's your terminal, the neuron, or the axon terminals. The axon is the yellow. This is an axon. This is a Schwann cell putting down its own myelin, forming a myelin sheath. So it's not really showing you the nucleus. The Schwann cells have a nucleus that should show up in every cell, a nucleus. But in between each of these Schwann cells and the myelin sheath, so Schwann cell and myelin sheath are basically the same thing. Like the cell has got a cytoplasm and a nucleus, but it keeps producing this layers of myelin. So these little nodes in between the myelin are called nodes of Ranvier, and that's unmyelinated spots. So this conduction could go really fast and jump over the Schwann cells or the myelin sheath. Again, it has to come up here first, sorry, to go into the cell body to be integrated and then continue down to the terminal, which is here, to the next neuron, which is sensory. I told you this is sensory, so this is afferent. But it happens on motor, of course, too, efferent as well. So myelination is not unique to motor or sensory. Both motor and sensory neurons in the PNS, most of them are surrounded by this myelin sheath that's made by the Schwann cells. Remember, Schwann cell is PNS, peripheral nervous system. All right, so here's more about let's look at the neuron, finally. The neuron itself, the nerve cell, the excitable cell, the membrane, the one that's amitotic. Right? This is a highly specialized cell. I think some people, when they saw it under the microscope or in the lab, said it looks like a kite, like a string in a kite, which is not a bad analogy. So look through the parts of it, and I guess these are the parenchyma, which is not only the structural but the functional units of the nervous system, not just structural by any means. They are functional, highly functional, specialized, because they don't look like any other cell, just like the muscle doesn't look like any other cell out of the 90 different cells we have. So here's the special characteristics. Like I said, they don't undergo mitosis, most of them, except we do have research, and there are some parts of the brain that have some ability to divide and reproduce. So lasts a person's lifetime. So like I said, once that brain is done growing, and then again, you're living with those brain cells. You just got to form new pathways. So you've seen the fontanelles, and don't get those wrong on the lab practical. I mean, fontanelles are spaces between the unfused cranial bones, which of course is important when you're going through labor and you have to get the baby's head out of that small birth canal under the pubic bone. But also, as your brain is developing, that soft spot, those fontanelles continue for a year or so, because your skull, if it fused at that point, your brain would continue to get enlarged and put pressure on the brain. So you need that little bit of flexibility for a year or two due to those fontanelles and that cartilage that's in between the cranial bones. Yeah. So cell body is like the master center of the neuron because it has the nucleus, has a cytoplasm, but the axon has a cytoplasm too. But the nucleus and the nucleolus is in the cell body. Remember the difference between the nucleus and nucleolus. Some people get that wrong on like a test or a lab practical, but it's pretty simple. Just the word looks the same. That kind of messes this up. So the cell body is also known as the soma. Remember, body is soma. I know the body of the vertebra is called centrum, but who cares, right? So soma means body as well. Perikaryon is like an old name that used to be given to the cell body of the neuron, but it's still, I like cell body. It's the easiest, right? Soma's cool too, but that's a general term. Cell body, use it, live it, love it, learn it. All right, and this is the biosynthetic center. That's where all the machinery is to make proteins, right? Like your rough ER, because the neuron needs proteins for structure, right? And chromatophilic is, that's your DNA, right? DNA and proteins and ribosomes. And proteins, usually histones or genetic, or they're part of your chromatin. So these are proteins that help build those chromosomes. But in a nerve, or in a neuron cytoplasm, they're called Nissl bodies. Like all this, it's like they call it chromatin in a regular cell or other cells. So all these substances together form like these dark staining histological parts called Nissl bodies. Look at the way that's spelled, Nissl bodies. I don't think we use that anywhere else, maybe one other place, but this is the name given to that genetic material that you see in the cytoplasm of a neuron, especially in the cell body. But it's not only in the cell body, but mostly in the cell body. So the big nucleus, right, that contains the DNA, and deep in the nucleus, you have a small little ping pong looking thing, ball, called the nucleolus. Now the nucleolus makes ribosomes through ribosomal RNA. Some pigments, melanin, right, within the neuron. Not sure why that's there, to tell you the truth. I don't think anybody really does. Look it up if you like. Good luck. All right, so again, the cell body is more of the receptive region, meaning that's where the incoming information is going to come into. So the axon goes out of the cell body. And you'll see another part of the neuron called the dendrites, which are really bringing information into the cell body. So you'll see, you have to really take a look at that. This is some terminology you need to get. I know they're given to you a little bit early, but let's look at this. And what you should also know that cell bodies, right? Cell bodies are what gives you the gray matter, right? Gray matter. So the axons are more myelinated white matter, but the cell bodies are gray matter. So clusters, listen to this. This is really, you got to remember this. Clusters of neuron cell bodies in the central nervous system are called nuclei. Nuclei is plural for nucleus. I'm not talking about a cell nucleus. I'm not talking about that. I'm talking that clusters of cell bodies are called nuclei. Like you'll hear of a name in the cerebrum, deep in the cerebrum, called the basal nuclei. And those are cell bodies that are part of neurons that create and secrete a neurotransmitter called dopamine. Right? So it's important when we get to the CNS that you know nuclei, we're not talking about the cell nucleus, we're talking about the cluster of neuronal cell bodies. Ganglia is the same thing except in the PNS. Right? When we're talking about autonomic ganglia. Right? That's what that first picture had, that word ganglia on it, but you didn't know what the heck that was at that point. So now at least you know what a cell body is. And a cluster of those in the PNS outside the spinal cord are called ganglia. So that's terminology. That's just some memorization, nuts and bolts that you have to go through. All right, moving on. So neuron processes, right? You have two of those. You have the dendrites, which come into the cell body, and the axon that goes out of the cell body. So every neuron just has one axon, but it could have different configurations. Dendrites could be multiple processes coming into the cell body. All right, and tracks are bundles of neurons in the CNS. And most of the time, tracks are myelinated. And they're both sensory and motor. Nerves, like I said before, is bundles of neurons in the PNS, and that could be sensory and motor, right? Most of the tracks go one direction. That's a good point. But nerves are bundles of motor and sensory, and sometimes autonomic motor and sensory too. So we really need a picture. I hope a picture's coming up. Yeah, finally. Damn. So again, these are all dendrites. Dendrite means like tree-like. Receptive means the information's coming into the dendrites from whatever, they could be receptors themselves, a dendrite. So let's keep this a one-directional process. Everything's coming in to the dendrite. Right? You can get information right directly to the body too, which is like from another axon, from another neuron. But again, most of the information is coming into the dendrite to the cell body, and all this is cell body. Right, and that's where you have the nissl bodies, right? I don't see that word here, but if you look, you really can't see it so much. I see rough ER, right? So chromatin, these little ribosome-looking things are going to be your nissl bodies or your nissl substances. The nucleus is the bigger membrane-bound organelle, and the little one is the nucleolus. So this chromophilic or chromatophilic substances like the rough ER would also include the nissl substances. That's pretty much unique to this. So the cell body, and now you have the beginning of the axon. So a hilum is usually an entry or an exit of a major structure. So the axon hillock is right where the axon starts. And right below that, you have the initial segment of the axon. The axon is all yellow that continues to our left to right. So in between, you gotta remember this, right in here, it's not an anatomical structure, but it's more a physiological area. And I like to call that the trigger zone. Because now we have to learn about this. We'll do it later, about depolarization action potential, right? So this trigger zone decides, it takes all the information that's coming in, whether it's excitatory, inhibitory, and you're going to learn that too. And if you get enough excitement into this cell body to this spot between the axon hillock and the initial segment of the axon to cause excitation, action potential, then the action potential will continue down the axon, depolarizing every segment of this axon till it gets to the axon terminal, right? And here's the axon terminal, which is going to secrete the neurotransmitter. Remember, acetylcholine, very similar. It doesn't have to be acetylcholine, but this very well could be a motor neuron we're looking at here. And this is PNS. How do I know? Because we're utilizing Schwann cells to manufacture myelin. So all these blue things are Schwann cells, which is really the myelin sheath. And the gaps in the myelin sheath are called Neuron. That's the gap. And yes, Valerie, you have to say it with a French accent, Neuron, in order to get full credit. Okay. So again, the direction of the impulse is going away from the cell body along the axon, but skipping over a lot of the parts of the axon to make it go faster to reach the axon terminals. So that's pretty much a typical neuron. And this particular neuron is definitely PNS because it's got Schwann cells. It's either most likely a motor neuron, which I believe it is, because if it was in the CNS, it would be an interneuron, and most interneurons, which are mostly in the CNS, are not myelinated. And they wouldn't be Schwann cells anyway. If it was in the spinal cord or the brain, it would be called oligodendrocyte. So get it right. Okay, so let's take them one at a time. Dendrites, again, dendritic means tree-like, like branching. So motor neurons, again, have hundreds of these for reception from one neuron to the next. And they're branched, of course. And sometimes you have some organelles in the dendrites as well, because it's kind of not all organelles, like the mitochondria. Like the neuron didn't mention the mitochondria, but there's mitochondria everywhere in here. Or maybe they did, I mean, I missed it. Just like any other cell, mitochondria, because you need to make ATP to get this conduction to happen, because you have sodium channels and potassium channels and sodium-potassium ATPase pumps. And then down at the axon terminals, you're going to have the release of neurotransmitter, which is exocytosis, which is an active transport that needs energy. So you're going to have mitochondria throughout the whole cell, basically, but most visible up in the soma or the cell body. So the dendrites are smaller, of course, but they do have cytoplasm. So a lot of incoming messages, like I was mentioning before, the incoming information are called graded potentials, meaning some of them are excitatory potentials, some of them are inhibitory. So sometimes you want to excite a neuron, sometimes you don't, and that's completely appropriate. So that trigger zone is going to take all these graded potentials, summate them, add them up, and see which one wins, the excitatory input or the inhibitory input. So if the excitatory input overrides the inhibitory, then you'll go ahead and have an action potential. All right, and we'll talk more about that. So I just want to introduce the word graded potential, because a graded potential is different than an action potential. An action potential, like I said in muscle, is all or nothing, like flushing a toilet. Once you put enough pressure on that lever, off it goes. It's not stopping, right? All right. So dendritic spines are just little processes off the dendrites, right? And they could have spiky or bulbous ends. And again, a dendrite could potentially be a receptor, because they are. Like, remember the free nerve endings you saw in the integument, that kind of jutting up into the papillary region of the dermis? Well, those free nerve endings are basically sensory neuron dendrites, the receptors themselves. Of course, it's not always like that, because the free nerve endings are more receptors for pain and temperature. But there's also, the corpuscles have enhanced receptors on their dendrites. And of course, in the special senses, you have very specialized receptors, like something like a rod or a cone, or the hair cells in the ear. And I can go on and on. Okay, so this is a microscope, electron microscope of a neuron. And you can see the cell body, and this most likely is the axon, but I can't really say. So of course I wouldn't put a picture like this on anything. Now the axon, also known as the nerve fiber, goes away from the cell body, and it starts right off the cell body in an area called the axon hillock. That is an anatomical name for that part. So some of them are really short, some of them are really long. Like you might have heard of the sciatic nerve, right? Oh, I had sciatica. I had pain down in my butt to my leg. So the sciatic nerve is a bundle of nerves. Bundle of neurons, both sensory and motor. So those particular neurons in a nerve like the sciatic nerve, which goes from your low back all the way to your foot, are very, very, very long axons, up to 1 meter long, like 3 feet. Yeah, like this one. Some axons, like the sciatic nerve, would be 1 meter long. And they're called fibers, the long axons. So some of them are very short, which you'll see different parts in the nervous system. And if the axons branch off to form new pathways, and that's what they're doing in certain parts of the brain especially, the axon have collateral branches communicating with other neurons. Like, think about, how your memory works. Something you could remember from a long time ago, a short-term memory, has different connections to different parts of your brain, like a memory of something you smelled or the memory of something you saw. And you put those two together, there has to be a connection somewhere to cognate that memory. So the axon collaterals put all that together, different parts of the brain that are cognitive of those different senses. So sensory is a good way, but of course axons are part of motor neurons as well. And it gets complicated too within the brain with moving certain muscles, or muscle memory in parts of the brain, which you're going to see. So axons branch at the end, and that's where you get the axon terminals, or terminal botons. I like axon terminal better for learning, because that's basically what it is. But sometimes I like another term for the axon terminal. Remember this in muscle, called the synaptic end bulb. I like that one too, because it kind of looks like that. It's the end of the axon, right? It's synaptic because it's going to forming a space between another membrane, right? Like in the muscle contraction, the neuron had a synapse or synaptic cleft between the axon terminal and the muscle membrane, or the sarcolemma. So functional characteristics, conduction, and the axon is the conducting region of the neuron. And the axolemma is the membrane of the axon. Now, the axolemma does not involve myelin. It's just the membrane of the axon. Because you're going to see later on, there's a membrane that's formed with the myelin, and that's called neurolemma. And that's much faster than using an axolemma to transmit this conduction. And then where's it going to end up? One direction, to the axon terminal, synaptic end bulb, or terminal boton. Okay, so the axon terminal stores in vesicles stores the neurotransmitter. Like acetylcholine is just an example, but that's the one you should know the best at this point. So the neurotransmitters that are stored in the vesicles will be released through exocytosis into the extracellular space. Well, let's call it what it is. It's called the synaptic cleft. And that's basically the gap junction between two neurons or between a neuron and the muscle. So here's the key. Look at this. It can excite, the neurotransmitter can either cause excitation of the postsynaptic neuron membrane, or it can inhibit it. So if it's excited, you're going to get yourself an action potential. If it's inhibited, no action potential. And what is this carrying on many conversations? I don't know what that means. Anyway, it's connecting with many neurons at one time is really what it's saying. There's going to be release to other neurons. So sometimes like one neuron could receive 100 different inputs, some inhibitory, some excitatory. And they could all be graded potentials that are sub-threshold. They don't have enough. Like you remember threshold from the muscle. It's very similar it's exactly the same actually in the neuron. Depolarization, action potential, repolarization. So axons rely on the cell bodies to renew the protein. So that's where all the machinery for making proteins is done. But the problem is, especially in the CNS, if a neuron is damaged, especially in the CNS, like the brain or spinal cord, it will just wither and die, and the proteins will build up and then become non-conductive. All right, so this is important to know. The damage of a neuron is usually irreversible, especially in the brain and spinal cord, in the CNS. No doubt about it. What about the axon? Transport mechanisms within the neuron or within the axon. So again, you're building a cytoskeleton. Cytoskeleton is the structure that gives it its shape, which is really important for a neuron, right? And this cytoskeleton is proteins, right? So there's motor proteins, which are really not that important to learn, but the proteins make up this cytoskeleton, right? So most of what we're talking about is antegrade conduction, meaning going from the cell body to the axon terminal, right? Movement of that. So that we have mitochondria all the way down. Cytoskeleton proteins, enzymes, of course, and your membrane components. Membrane components are your biphospholipid layer around the neuron, same as any other cell, but the membrane proteins that are embedded could be receptors or they could be enzymes. So you got to go back and think about when we did the cell, chapter 3, about the membrane of the cell and what it's made of and what membrane proteins are. They could be receptors, they could be enzymes, they could be channels, they could be carriers, they could be transporters, because most of the membrane is lipid. So some of the substances that need to work on a neuron are either hydrophilic or hydrophobic. So the proteins, of course, are hydrophilic. So they assist in having the hydrophilic molecules like proteins, carbohydrates do their job because they're polar, they're hydrophilic. But lipids are non-polar, hydrophobic, but of course they're lipophilic if they're lipids. Retrograde is coming back the other way, right? Which could be a problem, because you have viruses that could live in your cytoplasms of your axons, right? And also bacterial toxins. So viruses, like you may have heard of, like the chicken pox, right? The varicella virus can live in your cytoplasms of your neurons. And when you get older, you could have a condition called shingles, which is kind of like an adult version of chicken pox, right? So now, of course, there's a vaccine for that virus. But polio, again, we still get polio vaccinations. Like what we have with COVID now happened with polio back in the early 30s, 1930s, right? Are we 2030? Not yet. So viruses can cause retrograde movement and destroy the neurons, basically. especially motor neurons with something like polio, and in chickenpox, it's more sensory, very painful, itchy situation with that virus. And bacteria, again, could get into these, not as common, of course. Like you get a tetanus shot, right? Tetanus shot for the bacteria that causes tetanus, which is a bad condition of tetanus, not the good contraction of muscle tetanus. But again, that bacteria can cause retrograde movement and destruction of the neuron. So we want anterograde from cell body to the axon terminal. Yeah, polio, rabies, right? Rabies, that's still around, right? You gotta get your dog vaccinated for rabies and get from the local raccoons around, right? Herpes simplex. Herpes is very similar to the chickenpox, varicella. It's the same virus, just a different strain. And tetanus, which is bacteria. This is bacteria. Okay. Research is underway. When you see that, we can't talk about it very much because we don't know what we're talking about if it's still research. But talking about how we can use some of these viruses that aren't bad, like there are other viruses in our cytoplasms that actually are helpful, that we're finding more and more about that. That's why we study so much virology, because bacteria we know can be symbiotic, which means work with our systems to help us with digestion and absorption of certain nutrients. And also part of your brain needs a good intestinal flora, believe it or not, with research. So viruses can be helpful as well. So there's more, of course, research that has to be done on that, but it's going to happen, right? So this micro RNA corrected genes suppress defective genes. So it's kind of like this M or microRNA actually helps correct the cells that have been infected by a virus, like the stuff that I just got shot in my arm today from Pfizer, right? Whatever. Okay, and then there's the myelin sheath, right, which is the whitish lipoprotein, or protein lipid, whatever we want to say it. It's for protection, yes, but it increases the speed. So it has a little insulation and increase in nerve transmission speed in meters per second. So myelinated fibers are sheaths, like wrapping that towel around your arm, and usually on the thicker, wider diameter radius axons. So of course, non-myelinated fibers, which you don't see a lot in the PNS anyway. Most of these are interneurons. So they conduct a little slower because sometimes you need time for integration. So you're not going to see a lot of myelinated or unmyelinated fibers in the PNS within the nerves. You'll see more myelinated fibers, sensory and motor. The non-myelinated fibers in the PNS are more about autonomic neurons, which doesn't make much sense, but still, that's just the way it is. So most of the PNS axons are myelinated, period. So we know about the Schwann cells. That's the cell that has the machinery to make myelin in the PNS. A jelly roll fashion. Okay, jelly roll. I like the towel thing, but okay. Don't like jelly rolls. All right, so there's an outer collar, perinuclear cytoplasm. So the outer part of the Schwann cell or the myelin is called neurolemma. Remember, the covering or the membrane of the axon is called axolemma. Neurolemma, check the spelling, right? Neurolemma, and that's peripheral outside the Schwann cells, myelin. Yeah, and the membranes have less protein because they're more lipid. That's why they're good electrical insulators, right? And they're good for binding. The proteins are good for binding the myelin to the axon itself. Again, you could have a look here, this cartoon. You can see the way the Schwann cell, here's the nucleus of the Schwann cell. You can't see the nucleus in the axon because it's in the cell body for the neuron. So the Schwann cell will go around the axon as it's producing myelin, and then it'll keep wrapping and wrapping tightly till you get that nice multi-layer of myelin, which is called the myelin sheath, around an axon. Nice visual. You still see the nucleus too. Here's a electron micrograph of the axon, and starting to get myelin there around here. It's cross section. And you see the axon has cytoplasm. This could be a mitochondria, it's hard to say. Pretty cool. So in the PNS still, Schwann cells, there's gaps in between the Schwann cells, like Valerie's going to say they're called nodes of Ranvier. All right. So there's no myelin in those little gaps between the Schwann cells. So non-myelinated fibers, thin fibers, of course, not wrapped in myelin, surrounded by Schwann cells, but not coiling. So you don't have to worry about that as far as Schwann cells not producing myelin. You've seen this before. And here's all the parts of the neuron. Know them, live them, love them. And then we'll talk about different nerves and brain parts too eventually. There's much more to know about the brain as far as anatomy, like for lab, like pointing out the parts of the neuron is very rudimentary, but when we do the brain, it's tougher. You have different views, and it's a much bigger of a word bank than in a neuron, of course. Right, so myelin sheaths in the CNS is formed by the cell called the oligodendrocytes. That's the glial cell, right? The neuroglial cell. 60 axons at once, so it's a little different, right? It's kind of like Spider-Man, like the microglial cell of the astrocyte does the same thing. But oligodendrocyte seems to do the same thing with the spinning the web of myelin around. So when you hear oligodendrocytes, of course, you think of myelin. And there is a gap there. For some reason, we don't call it the node of Ranvier as often as in the PNS with the Schwann cells. And there's no neurolemma either. So it's really just helping organize what's going on in the axolemma. So that's something important right there. The neurolemma is about the myelin sheath in the PNS made by the Schwann cells. Yeah. So you should know this, the white matter are regions of the CNS, which the CNS is the brain and spinal cord, with heavily myelinated fibers, and that's white. And tracts are bundles of myelinated fibers coming in or going out. So coming in is called afferent, which are usually sensory. Going out is efferent, which is normally motor, right? And you could have sensory tracts and you could have motor tracts. And they're specifically named too, and where they go and where they wind up. So we'll revisit that when we do the PNS specifically. Now we're just going to learn about nerve tissue for a while. Gray matter is unmyelinated fibers, but mostly cell bodies, right? Gray matter. Like our cerebral cortex that I keep talking about is gray matter, right? Gray matter. Because it's cell bodies. We've seen this, the oligodendrocytes, forming the myelin sheath around the CNS cells. So remember, oligodendrocytes, CNS. They have gaps too, but we don't call them node of Ranvier. Now the structure of the neuron basically gives you the name of it, and it'll tell you which neurons are important for what and the type of neuron. So there's multipolar, there's bipolar, and unipolar, based on what they look like and the way the axons are set up with the cell body. So the one we've been looking at all along is the multipolar, you know, with the dendrites, cell body, then the axon going away. So there's one axon and many dendrites. Most common is multipolar, and these are motor neurons and interneurons. Right? Bipolar kind of have the cell body in the middle. So one axon and one dendrite with the cell body right in the middle of that dendrite and axon, kind of separating the two. So these are more for special senses. not really somatic senses. So special senses are kind of easy to remember. There's 5 special senses. There's vision, right? Number 1. There's hearing, which is audition, 2. Then you have vestibular, which is balance, also from your ear. That's 3. And then you have olfaction, which is smell. And then you have gustation, which is taste. So the retina is part of the eye, the neural layer of the eye that contains your rods and cones, which are receptors that have bipolar neurons attached to them. Okay, and same thing happens with bipolar neurons in olfaction and gustation, vestibular, and audition. So hopefully we'll see a picture of that coming up soon before we take a break. Okay, so the unipolar, or sometimes they're called pseudo-unipolar, but why not call them unipolar? Why not? So these kind of have like an axon that comes in, then you have a cell body, and then the axon continues. And that's really pseudo-unipolar, or we call it unipolar. So it's got a distal, say the receptor is here, like a dendrite really. And this is the part that goes into the CNS, coming this way, like the axon terminals on this end. Okay, and go either way. But this is unipolar, and most of these are somatic sensory. I'll even say somatic sensory, but it can be autonomic too. So these are your typical sensory neurons. Okay. So here's the picture. Here's your typical multipolar neuron. Right? I like this because, oh, look at the word, trigger zone. All of a sudden they're using trigger zone in the PowerPoint. Cool. So here's the axon terminals. These are the dendrites. Here is a cell body. This is a bipolar. So the cell body is in the middle. Here's your dendrites, or to the receptor. And here's your axon terminal. Right. This is a unipolar, which is sensory. Right. You have the receptors here. Trigger zone is up there, though. Look where the trigger zone is in this case. Kind of strange, right? Because it's near the receptor. Here's your cell body in the middle, and then your axon terminals here. So remember, the multipolar motor or interneuron, usually the motor neuron is myelinated where the interneuron is not. The bipolar neuron is more your special senses. We went through those. And your unipolar or pseudo-unipolar neuron is for your sensory neurons. Okay, good pictures. And again, you have different variances of those, and we're not going to really go through these. Just to look at them, you know, a Purkinje cell is important in a place called the cerebellum. Now, we didn't even mention the cerebellum yet. That's part of the brain, not where you do thinking. It's the little cerebrum. The cerebrum is where you do thinking and volition. And then you have a pyramidal cell, which really are interneurons within the brain connecting parts of the brain. And here's your bipolar, like your olfactory cell. They go through the cribriform plate, right? The receptors come up from the superior nasal concha through the meatus, and then through the cribriform plate to the olfactory bulbs, which sit right adjacent to the crista galli of the ethmoid bone. All right, so the crista galli, of course, you're going to see sooner or later a cribriform plate on the exam when you do that this week. The 11th, right? It's 11:59. So here's the retinal cells, again, bipolar. And here's a unipolar neuron, right? Reception. Here's the cell body. Now, if you have a group of cell bodies for sensory neurons in the PNS, it's called and it's in the back, it's called the dorsal root ganglion. Because that's a group of cell bodies in the PNS. Remember, you studied you memorized the definition of a ganglion versus nuclei. All right. So here's the 4 classifications, functional classifications of neurons. And this should be no surprise. Sensory is afferent, towards the CNS, afferent. Almost all the sensory neurons are unipolar. Cell bodies are in the PNS in a place called the ganglia. So really there's only a dorsal root ganglia. It's in the back of the spinal cord. You'll see that when we do the CNS. Motor is efferent, bringing the impulses, conduction, and however you want to say this, action potentials, from the brain and spinal cord to an effector, right? And pretty much all motor neurons are myelinated multipolar neurons. And most of the cell bodies are located in the CNS, except autonomic, which is a whole different ballgame. Because, the cell bodies start in your cerebral cortex, of course. Interneurons, or integrative or association neurons, are usually between the motor and sensory in the CNS, of course. So these are mostly in CNS. I like that shuttles the signals, right? And most are within the CNS. And interneurons are mostly multipolar neurons. That's a lot of interneurons, by the way. Some of them are so short that almost seem insignificant, but they still are integrative. So 99% of the body's neurons are interneurons, some wiring. Yeah. All right, so here's some more examples. You have the multipolar, which is a motor neuron. This is the spinal cord here. And this is anterior. This is posterior if you did a cross section. So the gray matter is really like that underarm or H. White matter is outside of that. So this is in a section of the spinal cord. And you can see the motor neuron starts in the spinal cord, goes out the front or ventral root, and then it forms a nerve. Actually, there's bundles of these go into a muscle. So that's somatic motor neuron, because it's going to a, say, skeletal muscle. That looks like a skeletal muscle. Well, the bipolar has the receptors in the retina, for example, the eye, and that neuron conducts towards the brain. And remember, that's the occipital lobe, right? Because that's what occipital means, the eye. So the cortex of the occipital lobe of the cerebrum is ultimately what cognates what you see and colors and all that. Right? Then the unipolar are sensory. So they're coming in, right, from, or say, a receptor, somatic receptor, like a pain receptor, dendrites, incoming, afferent, through the nerve. This is a nerve. And then the dorsal root coming into the spinal cord, which, remember, the spinal cord is CNS. And out here you have that unipolar cell body. There's not just one, there's a bunch of them. That's why they call this the dorsal root ganglion. And we're going to talk more about that, because we're still just talking about neurons. I just like to throw words at you. So the more you hear it, the more you remember it, hopefully, because there's going to be a lot of questions on nerve tissue on the next exam after the one you're opening up this week. Okay. How do we do with time? Action potential. Again, we have a pretty good idea what's going on here. So let's talk about depolarization and resting membrane potentials, action potential, repolarization, and a little bit about hyperpolarization. So let's start with the resting membrane potential. And later on, we'll just call it the RMP. So really, a neuron and nervous system as a whole needs to get back to this membrane to set up another action potential. It's very important, right? You have to get there to have another excitation, and a nice clean one and an efficient one, because the nervous system is controlling everything. So just to reiterate, like what we always talked about, the inside of the cell, and this is a neuron now, the inside of the cell has a resting membrane potential of minus 70 millivolts. It's very negative, right? All the sodium is on the outside. So the inside contains those large protein anions, which give the inside of the cell that negativity. Has nothing to do with the potassium being positive inside. So that doesn't add to the membrane potential. It just helps repolarize, which you'll see. So again, polarity is opposite charges, right? Like a positive, like say this is the membrane right here. So the outside of the membrane is positive in the resting membrane potential. So this is the ECF, or the extracellular fluid outside the cell. And the inside is really the cytosol, or intracellular fluid. So the cytoplasm of the neuron, axon 2, and dendrite, and cell body. So that's very negative. So there's an attraction, there's a potential for excitation, but it's polarized, meaning the pole on the outside is positive and inside is negative, like the North and South Pole, right? The pole, complete opposites, and they create a magnetic field. So again, they're opposite charges, and this is the RMP, resting membrane potential, which again, in a neuron is minus 70 millivolts. Very negative. So there's potential energy, and this is what makes it excitable, how it's returning to that resting membrane potential, and a lot goes into it. So again, the voltage, we're talking about millivolts, and that creates the potential energy. So that difference in positive and negative creates the potential energy. So large proteins, now we're not talking about inside the cell, we're talking about the ion channels, right? The large proteins are in the membrane, and they serve as ion channels. So they open and close based on voltage changes. So you're going to have potassium ion channels, and you're going to have sodium ion channels. Okay, and sometimes there's both. Sometimes you have symporters or antiporters where they're using the same channel. So there's two main types of ion channels for sodium and potassium. One is a leakage channel. So this works on concentration gradient, especially with potassium. So they're always open because they're not gated. There's no regulation to these. Again, if this is the membrane, right, and potassium's inside the cell, and it's more abundant inside the cell, there's a channel, a leak channel for potassium. So there's a lot of potassium on the inside of the cell compared to the outside. So there's just maybe a couple of potassium here. So because of simple diffusion, the potassium tends to leak outside the cell. And of course, that's going to make the inside more negative, and hopefully help maintain the resting membrane potential. That's really why these leakage channels are there, especially for potassium. There's not as many sodium leakage channels, because if you have sodium leaking in, that's going to cause maybe an inappropriate depolarization or make the membrane too jumpy, which is really a problem, especially in the heart, you know, with those membranes for depolarization, cardiac muscle. So there's more leakage channels for potassium. Gated channels, now again, proteins within the membrane that open and close due to sometimes chemically, like a neurotransmitter. Sometimes it happens through mechanical, right? Pressure, like on a dendrite. And another way is voltage change along the membrane, which usually comes from a stimulus. So chemically gated is basically ligand gated, the ligand being the neurotransmitter. Voltage gated is about the change in polarity and the change in voltage. So they'll open, these gates will open or close due to voltage. These will open or close due to the presence of the ligand, which is usually a neurotransmitter. Now, mechanically gated, when the actual shape is changed, like if you're talking about a touch receptor and you want to feel something with your thumb, you know, sometimes you have to put a little pressure onto those receptors to feel what's underneath your thumb. So those channels are mechanically gated, more to pressure and sometimes vibration, where chemical is more about the neurotransmitter binding. So here we go, chemically gated. And the ligand is the neurotransmitter. And the channel, now a chemically gated channel, one thing you should know here, that a chemically gated channel needs a receptor. And the receptor is specific for the ligand, which is a neurotransmitter in the nervous system. And there's many neurotransmitters, and we'll talk about a bunch. But so far, we've talked about acetylcholine a lot, and maybe we'll start to mention epinephrine or norepinephrine. Basically the same thing, epinephrine and norepinephrine. Same receptors anyway. Now, voltage-gated channels, the gates will open or close in response to change in the polarity or change in the membrane potential. Right? If it starts to depolarize, that'll open up channels in different segments, because remember the conduction depolarization goes along the axon, segment at a time. And then mechanically gated channels respond to deformation, like deforming the receptor shape, like putting pressure or feeling pressure or vibration, right? Will deform these microscopic receptors and cause the channels gates to open or close. Gated channels. So here's an example of chemically gated ion channels. So they have a receptor, and that's that very specific receptor, say for acetylcholine. So the gate is closed because there's no ligand, there's no acetylcholine available to attach to that receptor. So the receptor is basically part of a chemically gated ion channel, or you could also say ligand gated ion channel. And the ligand is the neurotransmitter, the chemical. So now we have acetylcholine present. Acetylcholine diffuses across the synapse or synaptic cleft, binds to its receptor, and then opens the channel. Now you see this is both for potassium and sodium. So this is an antiporter channel. So sodium could come in, and again, it's still against this, it's with this gradient now, so it's going down away from its gradient or downhill. So it's easy to get sodium to come in. That starts the depolarization of the inside, because here the inside of the cell would be negative with no presence of the acetylcholine or neurotransmitter, positive on the outside. But now that sodium comes in, you get more positive right there inside. This is sometimes to action potential. So voltage gated, again, you see no change in that membrane. It's still polarized. So sodium channel gates are closed for sodium, so it doesn't enter. You can't depolarize it until there's a stimulus. But if a stimulus affects the membrane, you start to get sodium coming in, then the channels will open. So that's a voltage gated channel. So sodium especially comes in very quickly. Those channels open up because there's so much sodium outside that you have very quick influx of sodium. And the influx of sodium is basically called depolarization, right? So it's going with its concentration gradient, like downhill from higher concentration to lower. It's basically a facilitated diffusion, right? So it could also go along towards the opposite charge too, as well. So that voltage polarity creates that attraction between positive to negative. So the voltmeter is what tests that number, the actual number. So the resting membrane potential inside of the cell, again, is minus 70 millivolts. So the inside, which is the cytoplasm, the cytosol is the fluid in the cytoplasm, it's very negatively charged. So massive difference in voltage. This is basically the change that could happen. You really don't have to memorize those numbers, but I would live here at minus 70 right here with resting membrane potential. So in that resting membrane potential, the membrane of the neuron is polarized. Polarized means two opposite ends, positive and negative. Negative on the inside, cytoplasm, cytosol, and outside is the ECF, extracellular fluid, and a lot of sodium on the outside, always, because this is the ECF. There's an ocean of sodium, all the ECF. Inside, again, negative along the membrane, mostly because of large protein anions, which are negatively charged particles. In this case, we're talking about proteins, which are inside the cell. And there's phosphates that are negatively charged. And you could have a bicarbonate ion that's negatively charged. So that creates a lot of negativity inside. So the potassium in small amounts on the outside, higher amounts on the inside, doesn't really add to the polarity in this case. But if potassium leaves, then it's going to change the polarity, making it more negative on the inside. So it has an effect when potassium leaves the cell. So repolarization is more about potassium leaving the cell, where depolarization of the membrane is more about sodium coming in the cell. And I'll repeat that later. So again, this is the difference between the ICF and the ECF. So the ICF is intracellular fluid, or the cytosol. ECF is the outside of the cell, the extracellular fluid. And of course, there's membrane permeability, which is also affected in a neuron by voltage changes, not just concentration gradient. Here's the voltmeter. So they stick a little probe into the cytoplasm of the neuron. Imagine how small that would have to be. And it gives you a number of millivolts. Voltometer, a voltimeter, a voltmeter. Many different names for that. So again, the extracellular fluid has a higher concentration of sodium on the outside than on the inside. Intracellular fluid means within. Inter means between. Intracellular fluid. Intra is within, and not inter. So get the difference between those, because that gets confusing sometimes. So we use ECF, extracellular fluid, for the outside, because sometimes that can be called the intercellular fluid or interstitial fluid. So we like to say extracellular fluid because it makes it easier to tell the difference between ECF and ICF. So now there's chloride ions, right? Chloride ions. But chloride ions are more extracellular, which you'll find out later. So I don't want to really talk much about chloride ions when it comes to the resting membrane potential, because the resting membrane potential is more about potassium leaving, right, and the sodium-potassium pumps than it is about chloride. So inside the cell, in the ICF, intracellular fluid, there's a higher concentration of potassium than there is on the outside. So potassium lives more in the inside of the cell, whereas sodium more outside the cell. And chloride does help balance those negative charges. Like chloride's more extracellular. So I'll say chloride is more ECF, where ICF is the proteins. That's in the ICF, the negatively charged proteins, which I like to call protein anions, because anion means negatively charged atoms or molecules. So potassium plays the biggest role in maintaining the RMP, resting membrane potential. And that's what we want to achieve, because then it's ready for a nice, clean depolarization action potential. So again, this is actually telling them what's going on pretty well. The large anionic proteins are inside. Now there's leak channels for sodium to go inside the cell, but there's not as many sodium leak channels as there are potassium leak channels. Like there's more potassium leak channels where potassium comes out by 25x, 25 times. So potassium leaves through facilitated diffusion because it's going down its concentration, from high concentration to low concentration, which is simple, doesn't cost any ATP. So the membrane is quite permeable to chloride, but again, we're not going to talk about that as much as sodium and potassium at this point. So this is important, the sodium, potassium, ATPase pump, right? Because what do we need ATP for? We need it for energy. And why do we need sodium, potassium, ATP pumps, or what do we need energy to move sodium and potassium? Because what these pumps do, like here's the membrane, right? And here's the sodium, potassium, ATPase pump. And then you have multiple of these pumps all the way through the membrane. So potassium is higher in abundance on the inside of the cell, and sodium is higher in abundance on the outside of the cell. So now in order to get the cell membrane back to its resting membrane potential, you have to pump sodium out against its gradient, right? And there's usually 3 sodium pumped out against its gradient, and you're going to pump 2 potassium back into the cell against its gradient. So both of these are going against their gradients. See, sodium's coming out against the sea of sodium. Potassium's coming in against a lot of potassium. So that's active transport. And this is an antiporter. So you need ATP. So what has to happen is you have to take ATP and you have to hydrolyze it. You have to use hydrolysis by adding water plus an enzyme, which is called ATPase. ATPase cleaves ATP into ADP plus this high energy inorganic phosphate. This I is for inorganic. And this is where the energy comes from to power this pump. So this protein installed in the membrane is actually ATPase. This is all ATPase. It's a sodium potassium ATPase pump. So it's readily using or hydrolyzing ATP to pump 3 sodium out, 2 potassium in. So that, again, because all the sodium that's outside, it gets it back to the membrane, resting membrane potential, minus 70 inside. So these sodium potassium ATPase pumps are the most important way of maintaining the resting membrane potential. Really important. And the potassium leak channels are also important. But this is everywhere on the neuron, constantly trying to maintain and get back to that minus 70 millivolts, which is the resting membrane potential. So now it's ready to be stimulated and depolarized. Whoa, this is kind of small. I can hardly see this. All right, so again, we're talking about concentration gradient. I don't see negatives and positives here. Sodium could pass through leakage channels, right? I'm sorry, potassium could go through leakage channels and come out. Sodium could do the same, right? Sodium could do the same, but there's, like I said, it was 25 times more potassium leak channels than there are sodium leak channels. And here's your sodium potassium ATPase pump. So this membrane protein is actually the protein enzyme ATPase, which when hydrolyzed, when ATP is hydrolyzed, will allow 3 sodium to come out each time against this gradient, active transport, and 2 potassium to come into the cell through active transport against this gradient. So that's a really important way of maintaining and generating the resting membrane potential. Nothing about chloride here, right? Nothing about chloride. So I don't want to kind of confuse you there. Okay. So membrane potential changes, concentrations of ions across the membrane change, and that's what's happening at this point. So there's 2 types of signals. Now this is really good that what your book is doing here. So there's 2 types of signals. One is a graded potential, which is incoming signals over a short distance. And some of these could be sub-threshold. And if you remember what the threshold was in the muscle cell, right? The threshold is a potential, like the threshold for a neuron is, or for action potential in a neuron is minus 55 millivolts. So these graded potentials never get positive enough to reach that minus 55 millivolts. So graded potentials kind of keep going, but over short distances, where action potentials are full depolarization, all or nothing, and go for long distances along the axon. Graded potentials can become or actually create an action potential if there's enough excitation above the threshold. So again, just to throw this at you, the graded potentials are not full depolarizations. They're just the beginning of depolarizations due to these incoming stimulus or signals, and they go over short distances, where action potentials are all or nothing, full depolarization to positive 30 millivolts. I think the muscle was like 40 in the action potential of the muscle membrane, but in the neuron, it's more like plus 30 millivolts, which is extremely depolarized, but that has to fizzle out. Like I said, flushing a toilet, all or nothing, where graded potentials can go on all day. So the changes in the membrane potential will now decide whether you're going to send the information to the next neuron or down the same neuron. So it's all about the electrochemical gradient. So here's really what you need to know here about the nuts and bolts here. So depolarization is the membrane potential becoming less negative. So sodium influx into the cell is depolarization, or causes depolarization of the membrane, right? Hyperpolarization is the opposite, where the membrane now could become, and of course this one's more positive, this one, the membrane potential can go down to say minus 90 millivolts. So now it's even more negative. And there's two ways that this can happen. One is too much efflux exiting of potassium, or influx of chloride, which is negative. So this is where we're going to introduce chloride. So if chloride comes into the cell, it's going to make it more negative and hyperpolarize the membrane. And that's going to inhibit an action potential, where depolarization could reach the threshold and cause an action potential. So sodium influx is making the membrane, inside the membrane, much more positive, where potassium leaving the cell can create hyperpolarization, or influx of chloride can lead to hyperpolarization. Now, hopefully the next page will tell you about repolarization, but it doesn't. So I'm going to mention, and repolarization is returning to resting membrane potential by increasing efflux of potassium, or just potassium efflux, because excess potassium efflux is going to cause hyperpolarization. Remember what polarization is. Polarization is opposite, totally positive on the outside, totally negative on the inside. So hyperpolarization is even more negative on the inside. Depolarization is turning the inside of the cell more positive. Remember, sodium is positive, right? And has much more effect than potassium on changing the membrane potential. So potassium is more about repolarization. Potassium efflux is repolarization. Okay? More voltmeters. So here you have a subthreshold graded potential here, right? You're getting a stimulus, but it's not reaching that minus 55 millivolts. So you're not getting an action potential. You're getting depolarization, but it's subthreshold. This one is the opposite. You're having, say, chloride coming to the cell, and the membrane is hyperpolarized. It goes down towards -85 or -90. So again, this y-axis is millivoltage. So the RMP you see is -70 millivolts. So this is dipping into hyperpolarization. This is the beginning of depolarization, but it's subthreshold. So again, this is just showing a small patch of depolarization where that stimulus will cause the sodium to come in to make it more positive at that segment. It's not telling you the whole picture of what's going to happen along the length of the axon, though. So again, it goes from segment to segment. In this case, there's depolarization. And you could kind of picture like if there was Schwann cells here on this axon, how it would jump here and then it would spread along the inside of the membrane, depolarizing it. Right, so this is depolarization, really sodium coming into the cell. So it dissipates quickly, which we'll see. So graded potentials don't always reach action potentials. So if they don't reach threshold, you're not going to get an action potential. So graded potentials could be either depolarizing or hyperpolarizing. So action potentials, again, these are not subthreshold. This means that depolarization went above threshold in positivity. So these are long distance communications, but they're all or nothing. They happen real quick and they have to repolarize. Yeah. So action potentials, again, don't decay. They keep moving. They don't fizzle out right away. They reach the action potential, keep moving down the axon. So this is the opposite of a graded potential, technically. So the action potential is what we call a nerve impulse, and that's what's excitatory from one neuron to the next, or one neuron to a muscle cell. And it involves opening voltage-gated channels, mostly for sodium coming in to depolarize the next segment or the next neuron. The key players are like this. Right? Here's your sodium channels, right, that open. And they could be ligand-gated, they could be mechanically gated or voltage-gated. This looks like a voltage-gated. So once this gate is open, the sodium comes in and it's depolarization. Then it's closed. It's inactivated by blocking the channel. And this is potassium. Again, potassium is higher in abundance in the inside of the cell. So it's blocked until repolarization, and then repolarization opens up the potassium channels and the potassium can leave, right, after a delay. So this should say repolarization, really. I mean, it means the same, meaning that originally you had the depolarization and that eventually went into repolarization. It has to repolarization. So potassium, I like this little thing. I don't know how I did this. Potassium coming out of the cell is more repolarization, where sodium coming into the cell is depolarization. So again, the resting state, RMP, all the gated channels are closed until you get a stimulus. So the leakage channels are open, so they just help maintaining the RMP. So activation gates are closed at rest, of course. Inactivation gates are open at rest, right? Causing the stoppage of the ions from flowing through those channels. So of course they're all voltage sensitive gates, especially the voltage gates. So, and it doesn't really say it here, but sodium channels are very fast channels, where potassium channels are slow channels. So sodium comes in much faster than potassium goes out. So sodium in, depolarization. Potassium out is repolarization. So they're kind of showing the steps here, like the resting state, where the activation gates are closed for sodium, so sodium's not allowed to come in. And also the potassium gates are closed. So you have a baseline of minus 70 millivolts. So upon stimulus, depolarization, sodium channels open and allow sodium to come into cell very fast, right? And then the inactivation closes after the influx of sodium. So the intracellular fluid becomes less negative or more positive because sodium is coming into the cell. So the threshold we're going to live is minus 55. So if the threshold goes from minus 70 after depolarization reaches the threshold of 55, then you can have an action potential, right? And throughout the axon, you're going to have positive feedback causing the opening of all the sodium channels along that axon. And the peak of an action potential, the action potential spike, is plus 30 millivolts, very depolarized, highly depolarized. So that's the spike, or the peak is plus 30 millivolts. So now the sodium channels open. In step 2, you have depolarization. So this is after the resting membrane potential. Now the sodium channels open and the activation gates are open. So allowing sodium to enter. Now potassium's closed during depolarization, potassium gates. So if you see up to 2, you see that rising from minus 70, minus 55, up to plus 30, the peak of the action potential. And then repolarization is sodium is inactivated, the channels are inactivated, and potassium channels open. So now the potassium exits the cell and makes the inside more negative again. We're going back to resting membrane potential. We're not hyperpolarizing unless excess potassium comes out or chloride comes in. So this is repolarization, is all about potassium leaving the cell and making the inside more negative, back to resting membrane potential, so minus 70 millivolts. So repolarization, the activation gates for sodium are closed, but the gates are open for potassium. So now as potassium leaves the cell, the inside gets more negative. So that's this part of the curve where you're coming back down to resting membrane potential. And right here, you have your possible hyperpolarization. But number 3 is showing you repolarization, which is efflux of potassium. So hyperpolarization is some potassium channels open. So more, or excessive is a better word, efflux of potassium, which makes the inside even more negative. So this could be like minus 85 to minus 90 millivolts of the membrane potential. So hyperpolarization is below the baseline, so more like minus 90. This is 70, minus 70. This is like minus 85 or minus 90 here. So that's the whole thing right there in another slideshow. All right, and shows you the whole curve. So this is kind of what we did in muscle. Numbers are slightly different with the minus 70 millivolts resting membrane potential, minus 55 millivolts is your threshold, and depolarization is your sodium in going up, peaking the action potential at plus 30. So the threshold is the threshold for action potential. That's all or nothing. Boom, it's flushed. And it can't stay up at plus 30, so it has to repolarize back down with the exiting or efflux of potassium. And sometimes it hyperpolarizes when too much potassium leaves the cell, so the inside of the cell gets more negative than the resting membrane potential. And that's inhibiting another action potential in a way. So again, the pumps help repolarize and restore the resting membrane potential, right? Okay, I think we'll stop on this slide. So try to remember what slide we are on next time, I will